Good morning, everybody, and I would first of all like to thank the organizers for providing that great opportunity for me to present my data here to the scientific meeting. And I have to say that I'm coming from a lab which has a prominent interest in the respiratory chain and its biogenesis. And we started then this project here on the respiratory chain remodeling in Barth syndrome. And I would like to give you only a very brief introduction into cardiolipin, of course. It is remarkable as it is considered as a hallmark lipid of mitochondria, as it is almost exclusively found in mitochondrial membranes and hardly no other cellular organelle. Here it is localized almost predominantly in the inner mitochondrial membrane. However, data in yeast have revealed that roughly a quarter of cardiolipin content might be also found in the outer mitochondrial membrane. Cardiolipin undergoes biosynthesis, of course, in the inner mitochondrial membrane, and this is also the place where it undergoes remodeling. And critically, this depends on the enzyme called tafazin, which is, of course, affected in Barth syndrome. Now, to understand what a defect in the remodeling of cardiolipin means for the physiology of the mitochondria, one has to know all the different pleiotropic functions which cardiolipin actually has for the mitochondria. And in this cartoon here, I just summarized a few of these functions, and one of these functions is connected to the biogenesis pathway of mitochondria, which, of course, itself is a very interesting process, as most mitochondrial proteins are actually encoded by the nucleus, need to be translated in the cytosol, and then they are transported across both membranes into the mitochondria, and this is mediated by these protein translocases, and they structurally depend on cardiolipin, which of course explains the role for cardiolipin for mitochondrial biogenesis. Another point I point out here in this cartoon is the role of cardiolipin in mitochondrial morphology, and this might be already due to its intrinsic characteristics of that molecule itself. However, there might be also many other aspects to this, and one I point out here is this recently discovered MyCos complex. It is a protein structure involved in the formation of these cristae structures. And what is interesting about this MyCos complex is that one of these constituents actually actively binds to cardiolipin, and this might imply a novel role for cardiolipin in morphology. However, I will focus today, as promised, on the respiratory chain, and this is due to structural and functional aspects of cardiolipin on the respiratory chain. The respiratory chain, of course, locates here in the inner mitochondrial membrane and in these cristae structures. And let me briefly introduce you the basic of this respiratory chain. It is a process describing the transport of electrons from the oxidation of metabolites, then via three complexes, 1, 3, and 4, onto its terminal acceptor, and this is molecular oxygen. Of course, during this electron transport, this is coupled with the proton export across the membrane, forming a membrane potential, which then is required for many energy-dependent processes. And one of them, of course, is the synthesis of ATP via the F1FO ATPase. Now, in our working model, these complexes do not simply float in the inner mitochondrial membrane. They rather form supercomplexes, and we call it a supercomplex when complex 1 forms a binding platform and makes a very tight interaction with this dimer of complex 3, and this platform then recruits several copies of the cytochrome C oxidase, or complex 4. And we know that these supercomplex formations are required for the efficient energy transduction within the respiratory chain. And we know that cardiolipin plays an important role in the formation of these supercomplexes. Now, this brings me now to the question I'd like to address here in these studies is how much are the supercomplex formations actually affected in Barth syndrome? And here I analyze now different model systems. The first model system is the stem cell model system for Barth syndrome, which we have published last year. This is a model where, first of all, I have to, of course, acknowledge the collection of fibroblasts from Ron Vandes. Then I have to acknowledge our stem cell experts in our at the University Medical Center in Göttingen, Kaome Guan, who was able to receive these fibroblasts and then transform into induced pluripotent stem cells. And they have done this with control cells, of course, in 3 different patient lines, all with a very well characterized mutation in interferon. And we, first of all, of course, performed the proof of principle experiment. We looked into the remodeling of cardiolipin in these cells. And these have been done using lipid extracts by mass spectrometry done by Fred Faz and Ron Vandes in Amsterdam. And what you can see, these are 2 representative plots for these cells. And what you see is that mature forms of cardiolipin in the Barth syndrome patient is, of course, decreased, where its precursor monolysocardiolipin is increased. So this reflects very well what has been also described for primary cells in patients, and therefore we take this as a good model to study Barth syndrome. And we then analyzed the respiratory performance, and this we have done by measuring the oxygen consumption rate over time. You can see here in black 3 clones of control, and in red 3 clones of one particular Barth syndrome patient. The first segment here actually is the analysis of the basal respiration rate, which is in our studies here quite significantly reduced. The next segment then shows the coupling after administration of oligomycin. This basically stabilizes the membrane potential, exerting an inhibitory effect on the respiration. And in the middle segment, we have here analyzed the maximal respiratory rates performed after administration of FCCP. This serves to uncouple the membrane potential and induces maximal respiration, which is also reduced in these cells. And the last is the proof of principle experiment, where we basically kill mitochondrial respiration via these inhibitors here. Interestingly, we saw this also for the other cells. They also behave differently in the severity of these phenotypes, and those seems to be even more severely affected, with a stronger decrease in basal, also with a strong decrease in maximal respiratory capacity. And it is, of course, noticeable that we obtain here different data compared to other studies. And I cannot completely explain that, and we would need to see whether our technical setup is basically similar. However, we then moved on and basically in these cells measured the membrane potential. We do this on isolated mitochondria, and in this complicated experiment, we use a fluorescent dye, which is basically quenched by the membrane potential of isolated mitochondria. And you can follow this probably in the black line here. After administration of mitochondria, this fluorescent gets quenched over time, and we can release this quenching by inhibiting the membrane potential using specific inhibitors. And if you follow now the red lines for the Barth syndrome patient mitochondria, you can see that this quenching is much less efficient, and therefore they obtain a much lesser or decreased membrane potential. We then analyzed the structure of the respiratory chain, and although this has been already today shown, I would briefly explain that technology once more to you. This is the Blue Native page gel electrophoresis, actually pioneered by Hermann Schegger in Frankfurt. And what this protocol means is depicted here. You start with cells, and then we isolate mitochondria from these cells. and solubilize them in a very mild detergent. And this entire protocol is designed to solubilize membranes on the one hand, but keep those complexes intact. And these native complexes can then be separated on polyacrylamide gel electrophoresis with smaller complexes migrating faster and these large complexes being retained on the top of the gel. And these separations we can use for Western blotting analysis, and we have done this here. Using antibodies against complex 1 visualizes then supercomplexes. Antibodies against complex 3 and 4 visualizes supercomplexes, but in addition also other complexes of lower molecular weight. And what is now interesting is to look differences between patient and control mitochondria, and you can see that in the patient mitochondria, supercomplex forms are either less abundant or completely absent. However, the respiratory chain does not disappear here. It seems to shift to a lower molecular weight, and you can see that here for complex 3, and you can see this also for this form of complex 4, where these forms are more abundant. Interestingly, a similar phenotype can be also seen for the other 2 patient mitochondria forms. The important experiment here is to basically analyze the stability of these respiratory chain components, and we have done this in the next experiment where we basically use completely denatured proteins in a simple SDS gel electrophoresis and Western blotting. And then we did this for a couple of constituents of the respiratory chain, like here complex 4 constituents, and here is a complex 1 constituent to prove that the amounts of mitochondrial protein of these constituents are identical in these samples. And this all summarizes basically to one basic picture where the respiratory chain in Barth syndrome patients seems to be stable, however, undergoes a remodeling in which supercomplex forms shift down to complexes of lower molecular weight, and this agrees also well with a respiratory deficiency in these mitochondria, as supercomplex forms are always considered as the major contributors to mitochondrial respiration. Then we also argued now in a different way. We would argue that supercomplex forms are necessary for the efficient energy transduction. So if you remodel the respiratory chain in patients, you might lose the energy. And this would be able to pick up by measuring the reactive oxygen species. And we have done this here in a very short time kinetic approach using this dye as a Ross sensor. And indeed, we're able to measure in isolated mitochondria from patients that here the respiratory chain or the generation of reactive oxygen species is significantly increased already in this short time measurement here. Next, we were, of course, also interested to progress this model a little bit further, and we therefore started to differentiate those cells into cardiomyocytes, and we are just obtaining the first set of data with these. And these is, again, a measurement of reactive oxygen species, and these measurements now agree better with what we've just seen before by William Poo, as measuring the basal respiratory activity here in cardiomyocytes of patients is now significantly increased compared to controls. They couple well after administration of oligomycin, and if we then apply FCCP to deplete the membrane potential and to uncouple, we are aware of a difference. Again, Tafadzin patient cardiomyocytes have a dramatically decreased maximal respiratory capacity. Of course, we are currently ongoing and looking into the structure of the respiratory chain in these cardiomyocytes. But before we had this cardiomyocyte model, of course, we were forced to basically look into our, or confirm our data, which we obtain in the stem cell, also in real cardiac tissue. And therefore, we turned our view onto this Barth syndrome mouse model. Of course, here we need to acknowledge Barth syndrome foundation. It is an inducible systemic knockdown of tafazin gene expression, and this works also in our hands very well in cardiac tissue. Eventually, these mice present with a severe cardiomyopathy. However, we look at this mouse model at a different perspective. We basically, also in collaboration with these cardiologists around Gerd Hasenfuss at the University Medical Center, we confirmed the published data that these mice also in our hands don't show any symptoms of cardiomyopathy. And then we basically look into this mouse model at a pre-onset state and wanted to describe which biochemical changes are actually ongoing here. First of all, we had to analyze the cardiolipin changes in this model, and here we analyze lipid extracts of cardiac tissue, again by mass spec in the lab of Britta Brügger in Heidelberg. And what these data revealed is a significant decrease of the predominant form of cardiolipin in cardiac tissue. And so we continued our measurements now, looking for the respiratory performance. And therefore, we had to isolate mitochondria again from this cardiac tissue. And as these don't show any consumption of oxygen over time, we had to feed in substrate, of course. Here we do this using succinate and ADP, feeding electrons into complex 1. ADP, of course, keeps the F1FO ATPase busy. And under these conditions, we at least see here dramatic decrease in the oxygen consumption rate. And of course, then the proof of principle experiment is inhibiting the respiratory chain with specific inhibitors. And we actually also, as these values always depend on the substrate combination used, continued these measurements using pyruvate malate, feeding electrons into complex 1. We also see somewhat a comparable decrease compared to succinate. And we also use this artificial substrate combination of TMPD ascorbate, directly feeding electrons into complex 4 and therefore circumventing the rest of the respiratory chain. And here, in opposite to the other data, we did not see a difference. We therefore concluded that obviously different complexes might be differently affected in this mouse model. And therefore, we performed, again, a different set of experiments. These are now these enzymatic activity measurements of individual respiratory chain complexes 3 and 4. We just used here the malate dehydrogenase as a point of reference, as this enzyme is an enzyme of the TCA cycle, obviously, and is not connected to the membrane, so it might not be affected by cardiolipin. And we saw a decrease in activity of complex 3, and actually basically, again, almost as expected from the measurements before, no defect in complex 4. So these were the functional analysis, and of course, these served as a basis for our structural analysis. And here again, these are Blue Native page analysis using complex or antibodies against complex 1 constituents, visualizes against supercomplexes, and antibodies against 3 and 4 constituents, visualizes supercomplexes. And these lower molecular weight forms, you can see there is a different band pattern as we are looking on cardiac tissue and not on stem cells anymore. And however, the general characteristic is actually very similar, as we see a dramatic decrease in these complex forms, in the supercomplex forms, and rather a shift to lower molecular weight, as you can also see this here with the monomeric form of the cytochrome C oxidase. And finally, I want to present you one experiment which is sort of summarizing the data on the functional performance of the respiratory chain, which correlates that with its position on the Blue Native page, and this is an activity staining. So basically, we use these separations in the Blue Native page and visualize enzymatic activity. We did this for complex 1 by the turnover of this blue dye, nitrotetrasodium blue, which basically leads to this precipitations in the supercomplex forms, much less in the knockdown mitochondria. And if we do this using diaminobenzidine, which basically visualizes the activity of complex 4, we can nicely see that activity associated with all these complex components from low molecular weight to the supercomplex form. And in knockdown mitochondria, this entire activity seems to shift down towards this monomeric form of the cytochrome C oxidase. Now, just to make this data set complete, we also, of course, looked into the measurement of reactive oxygen species over time, again, using in a short time kinetic approach this dye here. And what we observed is a somewhat increase in the reactive oxygen species in tafazin knockdown mitochondria. So when we summarize these, I presented to you basically two model systems. One is the human stem cell system, and the other one is the tafazin knockdown mouse model. Again, before the onset of the disease. In both cases, actually, we see a dramatic reorganization of the respiratory chain, and this has two consequences. One is a decreased respiration, and the other one a somewhat increase in reactive oxygen species. And with that, I'm coming to the end. I would like to thank, of course, every member of the Peter Rehling lab in Göttingen, also our immediate collaborators, Caro Meguan and Gerd Hasenfuß. Further on, we would like to thank, of course, Ron Wanders and Fred Fass, as well as Britta Brügger for their support. And I would like to thank you for your attention.
A very interesting talk. Thank you. I have a question about culturing of these iPS cells, because did you culture in the glucose, or it was in presence of glucose, or it was any other sugar?
So that was in glucose.
Okay, because that makes sense. Then what Bill's cells were in the galactose, so you have probably to have challenged these cells with a with some kind of metabolic stress to unveil this basal respiration phenotype. Yeah,
that would be a very interesting thing to do after we saw all these data. Yeah.
Yes, in connection to that question, there's actually a much easier way to explain these differences, that because stem cells or stem cell mitochondria are bioenergetically very different from mitochondria in differentiated cells, right? So there is this way, I mean, at least, I'm not sure this is true for induced pluripotent stem cells, so that's perhaps something you could answer, but it was shown that stem cells have an open megapore, for instance. I mean, this changes the entire bioenergetic function completely. And this could easily explain your differences, right?
Yeah, but we obviously, I mean, we have to try that with our own hands, but it seems at the moment that the medium and galactose and glucose as a growth, as a carbon source seems to also play a huge role.
So thank you. That was a very excellent presentation. So now as we're pushing so that you see excessive production of ROS detected as the elevated response from DCFH. So I wanted to mention to you that this DCFH fluorescence is actually the result of two potentially changeable variables. One of which will be exactly hydrogen peroxide, and that's what you imply. But the other one will be a metal catalyst of its oxidation. DCFH, in the absence of metals, will be stable like a brick. It will not fluoresce.
Okay.
Okay. But if you have a metal catalyst, then you kind of push its oxidation. So when you do these measurements in cells or mitochondria, the real change may be change of both of these factors, both the amount of hydrogen peroxide and the amounts of catalyst. And actually, there's a higher sensitivity to the catalyst content rather than to hydrogen peroxide.
Right.
So having said that, I also wanted to ask you, so, okay, let's say that you have elevated levels of ROS in these mitochondria. So, of course, it's probably reasonable to imagine that all three factors, decreased amount of cardiolipin, elevated levels of hydrolyzed cardiolipins, including mono and dilysocardiolipins, and finally possibly oxidized cardiolipins, will all be contributory to changed mitochondrial functions.
Yes, absolutely.
Okay, so if so, there has been old work decades ago, so that oxidized cardiolipins indeed impair mitochondrial functions. This has never been done with contemporary respirasomes and so on.
Yeah.
Okay, do you plan or have in mind, or have you done anything where kind of you take these test deficient mitochondria or cardiomyocytes and test whether, or titrate with oxidized cardiolipins, mitochondria certainly will be better, be able to affect the electron transport functions and so on?
That is, of course, an excellent suggestion, and for sure we haven't done that, yes.